Yes, actually, I was telling you about Mr. Tanmay. He's from Nagpur and he's pursuing veterinary sciences, and it's his 4th year. And he will explain us about RT-PCR test. I was telling yesterday, probably. Of course, I have selected the different topic, but I was telling that if he will be there, then definitely we will discuss on the same topic if it will be possible. So
Okay.
I think he can have a deep knowledge about RT-PCR test. That is why even I requested him, and finally Tanmay joined, and I am really very thankful for Tanmay that he
I
joined.
think you have more knowledge than me. Hello.
Really,
Coronavirus teach everyone microbiology to whole
yes,
world.
biology. Everyone can understand easily that the pores of a mask are not sufficient to avoid infection of COVID-19. Yeah, only snatching and other things can be stopped with masks, but not COVID-19.
If there was no COVID-19, we are not discussing about reverse transcription.
Yes. Okay. So obviously we will discuss about reverse transcription. And so Tanmay sir, one second.
Yeah.
First, let me ask Nikhil sir. Sir, should we start Tanmay so?
Yes, sir. Where is Siddharth, sir?
I don't know what happens to them. Siddharth sir is not joining today. Siddharth sir, Titan Saha, Himanshu Yadav. What happened to these guys?
Titans means in dental also.
Dental?
Oh, yeah,
Yeah.
yeah. He sent a message.
Okay, once again, once again. Himanshu sir is here.
Yeah, he is in dental clinic.
Hello.
Okay, it's okay. It's okay, Himanshu sir.
Yes, sir.
Yeah, we have Mr. Tanmay with us. He's from Nagpur. He's pursuing veterinary sciences. He will explain us about RT-PCR test. Sorry for today. I am not going to discuss about Veer Sehwal. What is Nikhil sir? What is the name?
Yes, sir. Lingayat.
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 Lingayat. We are not going to discuss today about Lingayats. We will discuss still on RT-PCR test because yesterday we deviated a lot. We were not focusing on the basic topic. So today Tanmay sir is here. So Tanmay sir, we have
There
with
was
you
a
Gautam sir.
okay.
Mr.
Hello, sir.
Gautam and we have Mr. Tanmay. He is going to explain about RT-PCR test. Yesterday you were not there. Is it? Gautam
Yes.
sir. Okay, so Tanmay so start.
Okay. There was just a breaking news before going to discussion that uh, price of reverse transcriptor PCR is just 400, and it will get the result in within 4 hours. And I think before vaccine is the biggest gift in 2020. But before vaccine, the RT-PCR is the most expensive and most beautiful gift to us, because this is the perfect test to detect coronavirus in any patient. The thermal scanner is not accurate, and only by this method, RT-PCR, we can uh, detect that coronavirus is present in our body or not. But we have to go through it. What is RT-PCR? There are three major words in that RT-PCR, that is reverse transcription and PCR. At, first, we have to do what is the transcription. We have to go through what is the transcription. That word include reverse transcript test. Without that word transcription, we can't understand RT-PCR. So transcription means uh, for our day-to-day life, we need protein to survive. And by the process of transcription, we make a protein. Gene make DNA, and from DNA, we get protein. And by the protein, uh, through the protein, every cells survive and humans survive. Uh, and for this process, DNA polymer is required for that protein. And that ribosome that you had studied in 8th or standard, third, fourth, and fifth standard.
In
That
class 10th.
in
Ribosomes, I
ribo-
have studied, but in class 9th and 10th.
okay, whatever it may be, that that protein is made in ribosome, and from DNA we get protein and protein structure, and the cell require protein. But in this case, DNA is converted into messenger RNA. That messenger RNA sends this message to the ribosome, and then ribosome made the protein. But now in this case, DNA and RNA is made from DNA. But when we have the sample of coronavirus, we have RNA single stranded. We have nothing to do with RNA, because RNA is a single stranded, and it's a very minute molecular structure, smallest unit of DNA, and it's very minute. So we have to convert it into DNA to study it further. And RNA is converted into DNA. In the first step, we discussed that DNA is converted into RNA. This is called transcription. But in
DNA
this case, hello?
is DNA converted into RNA or
Yes, yes, sir. Please continue.
separately?
DNA is converted into RNA is called transcription. But in this case, if we have to study coronavirus RNA, we have to convert RNA into DNA. So this step is called reverse transcriptase.
Okay.
And for this process, just like every chemical reaction, we need enzyme for the product. This required also product. For RNA converted into DNA, we require a reverse transcripted enzyme. So that this process is called reverse transcripted. And now the second word, PCR, that is include. PCR is the machine. PCR is the machine which extract RNA from the sample that is collected from our nasal swab, and in that nasal swab there is a mucus where that if RNA is present in that RNA or coronavirus is present, we collect the mucus from that patient. That mucus then we collect in a test tube, and that test tube we get sent to the PCR machine. That PCR machine convert that first we convert before we get enter into the machine, we convert RNA into DNA by the process called reverse transcriptase, and enzyme reverse transcriptase is itself. Then we multiply number of DNA. I am giving example why we do PCR. PCR is just like a Xerox machine. I am giving example. We have to study single RNA, and it's very difficult to study single RNA. So we made multiple copies of RNA to study that RNA. RNA, and then RNA is difficult to study, so we they converted into DNA. We have after the PCR done, we have the millions of copies of DNA that are easier to study, and we have many samples of DNA. So we convert that into DNA just like Xerox machine. And I am giving just example of one ant. Ant, ant chitty. Hello, hello.
Yes, yes, yes.
Yeah.
We can't see a single ant moving through the ground, but if there is a
Yes,
bunch of ants,
yes, they make a pattern.
they make a pattern. We can
Even
easily see
millions.
that. We can easily see that flock or bunch of that ant, but single ant we can't search. So just like this, we make a couple of or many samples of that coronavirus to study. Now, what does corona contain and which thing we detect? Coronavirus contain SEM protein and ORF 1A protein. So this is going to difficult. This is the structure present of this is the structure present in the coronavirus. Just like gatekeeper, I will give the example in simple language. Coronavirus have many spikes. Did you see in the structure? Hello, hello,
This is affected by what
hello,
cock?
hello.
Yes, hello, sir. Hello?
Audible?
Yes, sir.
Yeah,
Sorry.
everyone listen you. It's
Hello.
You are
That
better audible.
you have seen the structure of coronavirus. That there are many spikes. Hello.
Yeah, everyone
Yes,
listening.
yes.
Wait.
Many spikes. There
Yeah,
are
there
produced
are many
spikes.
So,
That
sir,
that's
sure,
why it's called crown.
there are many
That is its
spikes.
name is COVID. Means corona means crown, like related to
Yeah,
crown.
that crown
That's why it's
contains
called corona, right?
that, that contain E envelope. That through that envelope, that coronavirus enters into our cell, and the protein that I am going to seeing ORF1A, ORF1B. These are the target site which we get through the PCR. PCR machine does that. We have lots of sample, lots of DNA in our body, but the PCR machine detect only that portion of DNA. Did you get it? Hello?
Yes, yes. Okay, okay.
That and now how PCR work I am going to tell you about short.
Wow.
Normal PCR, and then I will just we have to reverse that process, so that will happen reverse
Okay, biology
transcripted.
student.
Yes, biology.
Okay, okay.
Yeah. uh, I will just first I will give the example of just like annual examination, university examination. From a whole book, teachers extract some important questions for annual exam,
Mm.
and then they make the copies of that exam, copies of that question, and then it delivered to you.
Yeah.
Okay, I am getting you. Hello.
Yeah, getting you. Fine.
Yes,
Just
yes.
like
We are
that.
getting.
Just like that, we are doing same procedure in PCR machine. We get the samples that
Messenger.
we are interested in. Hello,
Okay.
and then
Hmm.
we extract that interested gene, and we
Okay.
make the number of copies of that gene,
Okay, okay.
and then we study that gene.
Mm.
And there is a one substance called primer.
Okay.
Primer means raw material that helps to detect this process. There
Mm.
are mainly three process, but one thing that ask in every exam in MPC or UPC also, that who discovered PCR. So first of all, I will to tell Carimulis discovered the PCR. Hello,
Yeah. So
that
who discovered?
When?
Carimulis.
Hello.
Hello.
When Carimulis discovered?
The time I am not sure. 1880 in the period of 1880 or 1890. I am not sure, but no, not date are mentioned. But I know only who discovered Caramelis. No one, I don't think no one will ask a date. But the scientist at Caramelis.
Oh, so
And
actually, he's a lawyer. That's why he's asking.
I don't know. I'm I didn't know about that. Sorry. Uh, And this is the most important question that every examiner should ask, or every examiner should get through it. And now I am going to describe a PCR. Polymerase chain reaction, and the word polymerase means enzyme that converted DNA into RNA. If we bring RNA in front of polymerase, then DNA is converted into RNA. And if we connect DNA polymerase, then RNA is converted into DNA. So here, coronavirus is single stranded, and PCR. What is the mechanism of PCR? PCR detect the antigen that present in that sample. First, we collect. Just I tell before you that we collect the sample, and if sample contain antigen, the antigen is detected by PCR. This is the main application. This is the main application of PCR, and PCR is also help in you to detect the cancer cell, because cancer cell have specific sequence, uh, and cancer cells are mainly the cell that stop dividing. So that was there was a tumor that some cells are grow uh, repeatedly. So there will be a swelling at just like what we say, there a big tumor in that specific area. So the cell or the sequence which are going to mislead, that are detected by that PCR machine. And PCR machine have one marker. So if someone have question that how that PCR detect that specific gene, the PCR1 marker, genetic marker that helps to know from that sample will that are present only in the coronavirus. We have already that sequence that are present in sequence means DNA. Everyone, just like we have our name Tanmay Katle. It contains TANMAY. Hello,
Yeah.
this is my sequence. Hello,
Yes, yes.
Yes, yes, yes.
yeah, this is my sequence TANMAY. Okay,
Okay.
and in
Yes,
the somewhere
Tanmay,
book,
Tanmay.
yeah, and in the puzzle we bring that 8 blocks in randomly. Hello, if we bring
Yes,
that
yes.
single TANMAY, just single word, we have to arrange them in a particular order. Hello,
Yes, yes.
Okay.
Then only I can call Tanmay.
yeah. the
Mm,
there is a particular sequence TANMAY, not Y first or not A first, but we know the sequence. Hello.
okay,
Right,
okay.
right,
Why?
right.
Unity description this word. Am I right?
Yeah,
Unity
the
groups.
just like my
Oh,
name have particular sequence TANMAY, first
okay.
T, then A.
So we
Then
have to do encrypt this word. Am I right?
hello.
So we have to do encrypt. This is your
Uh,
Yes, it's
description.
like encryption. Right.
Okay. Okay.
same have coronavirus at some particular sequence, just like I
Okay.
have sequence of TANMAY. That we
Mm,
have to detect that sequence,
okay.
and we do done through PCR. Are you
Oh,
getting or not?
yeah, getting
First
it.
of all, tell me. Hello.
Yeah, getting your point.
You
This
know
is
you know DNA. You know DNA.
yeah, I know
DNA
DNA.
has
When I study in
DNA,
school.
there are many, just like staircases, there are many lines between DNA. That spiral DNA. In that spiral DNA, there is a particular sequence, and everyone has that sequence. RNA also, coronavirus also have that sequence, and we detect that sequence, and we extract. Then from extract, we first we done extension of that small sequence, and from extension we replicate it, and after replication we do millions of copies. I will just
Okay.
tell example what PCR do. PCR are mainly three processes. um Hello,
Tanmay sir.
denaturation.
Tanmay sir. Tanmay
Hello.
sir. Tanmay sir. With
Yeah.
Tanmay, so of your screen sharing.
Hello.
Yes, can you
Hello.
suggest us with with Tanmay, so means with the screen sharing on the
Yes,
internet
sir.
if you get
Screen
somewhere?
sharing or
Yeah, yeah,
a image, it's
just
very helpful for
yeah,
us.
yeah, I
It
will.
will be really
Because
helpful.
it's too much,
Yeah, yeah,
yeah, complicated.
yeah. Should I
And
s- sh-
that's why he was saying, Nikhil sir, that's why he was saying that I'm going to so make you Tanmay so.
Yeah, first step is so.
Yeah, now we are watching Tanmay so, and we are
uh,
having a little bit break. Meanwhile, Tanmay sir is searching the topic
Okay.
better
uh,
than.
how should I
Mm.
share this? I have to
Sir, option is
Yes,
right side to
yeah, yeah, yeah.
means call cut button. Yeah.
Okay, okay. Did you visible?
Yeah.
absolutely visible.
Oh, don't watch my history.
No, 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 no.
I will show you diagram. First of all, I will show transcription.
छोटा छोटा दिख रहा
छोटा छोटा
अरे
दिख रहा
बाप
है
रे. Yes, Siddharth sir,
है या सर
please zoom out.
Okay.
बड़ा
In
बड़ा दिखेगा.
yeah, when we zoom out, yeah, means
Yeah, I will.
alphabet is not clear. Sentence is
Yeah,
not clear.
maybe. Right, right.
I will show. Now I will. We will study
Blur,
PCR. Okay.
blur sentence.
Hello.
Yes, now Sumit.
1 second.
Yes, yes.
Where is my history? RT-PCR.
And now your mobile is very dangerous.
Yeah.
I think
First of all, I will
in
tell what is poly. Yeah, everyone getting?
Yeah. PCR.
Yeah, polymerase chain reaction. I already tell it is a technique used to make multiple copies of DNA of interest.
Okay. Okay.
I already told you that the gene of interest, that means gene of corona, I mean RNA, is our of interest. Okay.
Hmm.
Hello.
okay.
Yeah.
Yes,
Everyone.
yes, yes.
First, we have to search it,
Oh,
that small RNA. Then we have to convert it into DNA. Then we have to make sample, millions of sample, and from that sample, we can surely say that the individual is positive or not, and that is
okay.
the that is what PCR. Polymerase mean enzyme, and chain reaction means we make multiple. In first cycle, we can make the copies 2, in second 4, in third 8, 16.
Okay.
We can make 30 cycles of PCR.
Okay. Okay.
Okay, and that will give 1 crore samples. Hello, that give 1 crore of sample. That video will show in the next. That here is the one. Let's, for example, there is a sample. Okay,
Hmm.
and then we extract DNA.
Okay.
And there in the green marker, there is a target DNA,
Okay. Okay.
just like target question by annual examination. Okay, then just like we make a list of important question, and then it copied, and then everyone get that question sheet. Just like that, we extract the target DNA that we are of interest. Then
Hmm.
we uh, that sample get collected into PCR machine,
Okay.
and there are 3 major process: denaturation, annealing, and extension. So that DNA, there is the in upcoming video we will study that. You will see that there is a copy of sample, large sample of DNA that are easier to study.
Okay.
Is there one lawyer student that you tell to me earlier? Hello, hello,
Yes, sir.
Sagar.
Yes, sir. Yeah,
Mr.
yeah.
Nikhil, it's a lawyer.
You in the Supreme Court or judge, the decision is given by not on the basis of only one. What will say? Not only one. What to say that?
Only one side.
Yeah. No, no.
Yeah, yeah.
Only one.
That here
Based
both
on one proof,
parties. Both parties. Yeah.
we have to take many proof. Not only
Yeah.
based on one
More
proof.
evidence required.
Yeah, more evidence. That evidence. Just like that, we need more sample. Okay.
Hmm.
Just like on the basis of not only one sample, we have required couple of samples. Just like couple of evidences. एक just so there is a thermocycler. The thermocycle, I will tell you that use of that thermocycle, it denature the DNA that we collect. Okay, the target DNA. Hello,
Yeah. Yeah.
target DNA. With the help of that thermocycle, we increase the temperature, and then we break the DNA. There is a target DNA. There is primer, raw material. This primer bind to the target DNA. And you are seeing that primer we used to, they are like complementary to each other. Hello, the
Hmm.
primers are complementary to the target DNA. Are you getting it? This
Yeah.
primer bind automatically bind to that target DNA. Okay,
Okay. Okay.
Mhm.
and that primer is of known. We know the primer sequence.
Hmm.
Hello, and if the primer bind to the DNA, that what we do? We do that. We collect the sequences of coronavirus in the primer. Okay.
Okay.
If primer bind to the DNA, then the person is COVID positive or RNA. And if primer not bind to the that sample, then there is no COVID-19 test is negative. Okay, get it? And hello,
Yes, yes. We are getting a little bit.
we collected in our test tube. Polymerase is enzyme. Okay,
Sorry.
polymerase is enzyme. Okay, the word
Okay.
first comes in the PCR polymerase. And now
Mhm.
you are getting that at least why we call that chain reaction.
Yes.
Just like a chain, we make a long samples of DNA. Okay, that's why we call chain. Just like chain reaction game, there is a. Okay, you are getting.
Like an atom bomb. We can say
Yeah,
corona bomb. Okay.
yeah, there is a chain. First step, target DNA. Okay, hello.
Okay.
Okay,
Yes,
there is a
yes.
target DNA. To know the primer to bind, we have to first break it. Okay, so we
Mhm.
are going to put the DNA into single strand. First, we decoil the DNA at denaturation 95 degree. First process of PCR. Okay, there are three: denaturation, annealing, and extension. Okay,
Mhm.
first we uncoil the DNA
Mm.
and then we denature at 95 degrees Celsius. So why that strands will go wide apart. That strand break of target side of only target side. Now this is the polymerase. Okay, tag DNA polymerase. And these are the primers. The polymerase helps to primer to attach the target site. We don't know the DNA of, but we know the primer. Okay,
Okay.
hello. We
Yeah,
don't know
yeah.
the
Yes.
DNA sequence, but we know the primer sequence because we add them, and we have that sequence. Hello,
Yeah, okay, okay, okay.
primer DNA is of unknown, but primer is known. And if
Oh,
known primer is bind to unknown, then unknown become known. Did you get?
What a mathematics.
Huh?
Nice mathematics.
Yeah. Did you get? Or I have to give example or
Yeah, once more.
yeah. A primer and DNA is complementary. Okay.
Hmm.
Complementary means opposite. They are just like puzzle that they attach. You play the game of bricks. We have to
Bricks.
fix it. Bricks.
Yeah,
We
yeah,
have to
yeah.
make a
Yes, yes.
wall.
Mhm.
Yeah, and that particular wall is going to particular bricks. Okay, not all of.
I got. I have also
Yeah,
played on keypad
yeah.
phone.
That particular side bind to particular block. Okay, here
Okay,
and
okay.
that, and you have seen many their strands, vertical strand. Okay, that strand contains sequence that we know. If that sequence bind to that upper strand, that then the upper strand and that primer contains same sequence. Hello.
Yes, yes.
Yes, if we know the primer contain the sequence of coronavirus, just I said TA and MAY, just like
It's a
corona.
simple. It's a similar to Tala Chabi Siddhant. Key lock. Key lock theory.
Okay, that coronavirus has sequence. That sequence are present in that strand. If strand matches to the upper strand that contains sample, then that person is COVID positive. If strand doesn't match, that
That person is
uh,
COVID negative.
negative. Okay,
okay.
Okay.
because they are similar, complementary. They are complementary.
Mhm.
Yeah.
Just like north south pole, north and south pole. Hello.
Okay, okay, okay.
South get attracts to the north just like, and if we have two ends, and if we know only one end south, and if north they don't if they get attached, then we can say other one was north pole. But if south we have no only one end south and other is repelling, then we can say that other was also south. Are you getting? Just
Okay.
like
Yeah,
magnet pole.
very fine. Okay,
If they
okay.
attract, we can, if we know one side, we can diagnose other side, and we can tell other side.
Okay, okay. So PCR mission already predefined normal DNA program. So once, am I right? If any other mismatch against the DNA, it automatically find out this is a target DNA. So,
Automatically find.
oh, okay. So in PCR mission already predefined normal DNA structures and algorithms, everything in PCR
And the
mission.
primers are pre-decided.
Okay, okay, okay. So if any DNA missing from that algorithm, automatically it will trigger
Primer,
this is the target
the
DNA.
primer means, yeah,
Okay,
yeah. That's why I'm saying the primer will automatically trigger
oh,
that point,
okay.
and that after that primer attach,
So it will never connect only the corona affect DNA. It will, if any cause,
any,
it will get enough. Any disease
yeah,
it will get ensure.
corona is become star now, so that's why I use
Oh,
corona. It will be
okay.
helpful for all viruses,
Find out
not
the okay, okay, okay,
all
okay.
bacteria, all
That's why I asked.
viruses.
Okay, okay,
Yeah,
okay.
yeah. Excellent question. Thank you. You can help me to explain other also easily.
Thank okay. Thank you.
Thank you. Thank you very
Thank
much. Uh, that now see.
That's all, Dr. Sahab.
Now see. Just there. I was just saying complementary, just like north test to the south. Here also same thing happens.
It
A
look like
binds to the
half
T,
moon.
T binds to the A, G binds to the C, T A. That's our complementary A T G C. You
Mm.
can see now.
Yeah, yeah,
Yeah, are you seeing? Just like north
yeah.
south poles.
Mm.
Yeah, they are binding. And we know the sequence T that primer T A C A A T C T G, and they bind to the complementary,
Mm.
and they are correct in equal direction. One from north to south, other from south to north. Just see.
Okay, okay.
This is little bit. This is the most difficult part in that poor whole PCR.
Okay,
But you
okay.
are getting that they are complementary. At least you can tell that they are complementary. Hello,
Yeah,
from the given slide, we can say that these are complementary. Okay. Now
okay.
we can from the sequence we can say that complementary sequence, and that's unknown DNA. We get sequence A T G
Mm.
T T A G A C. Did you see it? Because of primer, we get that sequence A T, and that's why we have get through the PCR. This is our main name A T G T T A G A C.
Mm.
Okay,
Okay.
are you getting or not?
Yeah, I'm getting little bit.
Yeah,
I
the
got an
sequence we
idea.
have.
What is a PCR? I got a little bit aware of PCR mission.
Yeah, I
What is
will
a
explain one more time. The DNA. You can tell me answer in yes or no. The DNA was unknown. No?
mm.
DNA was unknown, but primer was
Yeah,
known.
yeah, okay. So
But
PCR
now
mission only
it is
converted
also known.
unknown object is to a known object. Am I right? So
Yes, that's
PCR
why I'm
mission
going.
work is unknown object converted in a known object with the help of some sequence weight.
Yeah,
That
that primer,
you
primer or
yeah,
raw
that primer.
material.
Yeah, raw material. Okay, okay.
First we know the only sequence of primer, but now from that
Mm.
complementary, because of that
Okay,
opposite, we get now at an unknown stand also.
okay, okay, okay.
Now we get that sequence, but now we have to multiply it. Next process is simple.
Okay,
We have to extend it by DNA
okay,
polymerase, the polymerase enzyme.
okay, okay, okay.
Polymerase enzyme already told you. That's why the machine have name polymerase. Polymerase helps to extend that sequence.
Okay. I know only polymerism. I never know only polymerism.
Yeah, polymerase. Yeah, this is
Yeah.
just
Okay.
this
If
is
I
just
want to know
enzyme.
fully polymerism, I have to study it once again in my biology class.
Yeah. The polymerase is just an enzyme that helps
Okay.
to that helps connect, or just like a glue.
Okay, okay,
Okay.
okay. Okay.
Yeah, now the strand get extend of known DNA.
Mm.
Now that unknown DNA become known due to that
Oh,
complementary strand
okay, okay, okay.
cycle. Now we have to just replicate it, the same process.
Mm. Okay.
Okay, sir. Thank you very much. Is anyone having still further questions? Yes, Siddharth sir, please ask.
What is his name? I didn't know his name also.
His name
Hello?
Tanmay, Tanmay.
No, this is a sequence of, I know. He
Hello?
said this is a sequence of his DNA. Hello, Mr. Tanmay,
Yeah.
what is your exact name?
What?
What's your name?
Tanmay.
Tanmay.
Tanmay. Okay.
Yeah. My name has particular sequence. DNA, each DNA have particular sequence.
Okay.
Yeah.
Okay.
Just like
Yes, Nikhil sir, please ask questions.
yeah, that now we can see that yellow portion is now extending that
Hmm.
target.
Yeah.
Let
Gautam
Nikhil sir
sir.
ask question. Nikhil sir.
Yeah, Gautam sir, you also a science student.
No, but up to 12th I have studied biology.
Okay, okay.
Up to 12th means you are a biology student.
Yeah, 11th and 12th I'm in first group. On the time I studied little bit DNA and little bit RNA.
Okay.
Uh-huh.
Now
You are a
the
science
formula
student.
for counting, it's like 2, 4, 8, 2 raised to power n.
Oh,
2 raised to power n.
okay, okay.
Siddharth sir, please ask question. Hello?
2 raised to power n.
Siddharth, one.
Mm.
Siddharth sir.
Yes, Siddharth,
Yes,
you can
yes.
ask the
Again.
question.
Sir, please ask. Siddharth sir,
Yeah.
please
Yeah,
ask.
in our blood, when any person suffer from coronavirus, so antibody will active. It is only in coronavirus cases or in our fever. If we have fever, so antibody will also active.
Yeah, if antibodies are prepared after 14 days of every, not only virus, every foreign particle also. If some foreign object enter in your body, antibodies are produced. And your question is that not only for not coronavirus, we have antibodies for all the viruses or bacteria. And if we not able, if your body is not able to detect that structure or virus, then we have no antibody, so there is suddenly death.
Okay,
Yeah, we
so
have
if we have
uh,
fever, so our
uh,
antibody will also active, na?
yeah.
So how can we find coronavirus? Because like that in radic, what was the name? Yeah, antibody that detector like that in only 1 hour we can detect coronavirus. So with the help of antibody we can detect. So if we have
Yeah.
any person fever, so antibody can also say I am here, right?
No, particular antigen or particular virus or particular antibody. Hello.
Okay, so
Particular
there are
particular antigen have particular antibody, and same antibody will produce in my body also, your body also. And if we extract the antibodies from the COVID active patient, the same body are present in our side also.
so there are
So
also type of antibody.
Yeah,
yeah,
several
there are
type of antibodies there
several types of
Okay.
in our
antibody,
body.
and it depend on antigen. Hello.
Okay,
Yes.
and what about like someone person who are not any symptoms, even he is suffering from coronavirus in
Hmm.
50%.
Yeah.
So what is the cases about it? Why they are not saying any kind of symptoms?
Yeah, because their body are capable of producing large quantity of antibodies.
Mm.
That's why,
So,
and so not all, because it's depend on your immune system. If your immune system is good, then your there are 4 types of cell: memory cell,
so
helper T
if
cell.
any
Hello.
persons have any symptoms and he is suffering from coronavirus, so they no
Yeah.
need any type of
No,
treatment, any type of vaccines.
no. No, they don't need any type of because their body are capable of producing antibody, and not all capable of producing, because it's depend on your immune system, how active your immune system.
Yeah.
If
Where
you have good immune system and good lifestyle, then it's depend on your immune system especially, and how your immune organs are developed.
yeah,
Okay.
somebody yeah,
Also,
somebody patient corona positive, then he take a vaccine. So what happened?
No. That's not happen. If someone have already have coronavirus and we are not injected them, we have just to cure them. We can't inject a coronavirus vaccine on the patient who have already infection, because it will increase the infection. Vaccine is what? It is only dead antigen, means dead coronavirus.
Yeah. Brahmanand
Yeah.
sir.
Hello.
Hello.
yes, sir.
Brahmanand sir,
Yes, hello.
Yes, sir.
virus.
Please
Hello.
ask question.
Actually, my question is that why people are get morely positive case in the rapid test? With the rapid
Uh,
test, why people are get more, yeah, as compared
Rapid
to the
test.
other test?
Because
Yes, in
because
rapid test, as compared to the
there
other
are
test, yeah.
because there are 19 types of coronavirus, not only one type of coronavirus. If you have coronavirus of 2002, you have only a little bit fever and sneezing. There are 19 types of, there are many more possible combinations of coronavirus, and the coronavirus is start replicating, and the country suffer from COVID-19. And if you have infection of coronavirus, rapid antigen directly show that infection of coronavirus, not COVID-19. COVID-19 means there are 19 types of coronavirus already existed.
Oh,
So 19 types, you have one of them in
okay.
rapid test. In rapid test, you have one of them. And if rapid test
See,
is done, there is a coronavirus, not COVID-19. And if you do
One thing,
RT-PCR,
one query. Tanmay sir, one query.
if, yeah,
Are you saying about COVID-19 means that 19 of 19 type, or
yeah,
this 19 is a
it is
different
of
thing?
19,
Yeah,
it is of
2019 or COVID-19 is a
Yes,
different thing.
it's a
Scientific thing.
2019. Yes.
yeah, it's also not COVID-19. I mean, it's of 19 means it's a 19 COVID-19 type.
Okay, it's a coincidentally, it's a fortunately the same digit. Is it?
Yeah, no.
Okay, okay.
That's I was given to you COVID-19, and there are many types of coronavirus, many
Okay, okay,
such,
okay.
and
I got, I
the
got.
Himanshu
RT-PCR
sir.
done RT-PCR done the recognition of COVID-19, especially, and the COVID-19 is most harmful. And if you have the infection of coronavirus that occurred from 2002, there are many more coronaviruses occur and gone. But this is the 19, it is mutated. It is different
As
from that.
one
Himanshu sir.
interesting thing. Yes,
Hello.
Himanshu sir.
yes,
Yeah.
sir.
Yeah, please ask question.
Yes. So, my question is, if one person uh, check up COVID-19, then uh, in one time he was positive, but in other day he was negative in 2 hours time. So why this is possible?
No, that was that negligence in test. That's why PCR is most demanded. Hello, PCR is
Hello.
90%. If you have done other tests, just like rapid test, antibody test, these are only 50% successful. They will give a 50% accuracy. It may be wrong or right. That's why RT-PCR we have done 90%. It has 90 to 95% accuracy, and that we have that are technical issue, not body or not that of
How the
antigen.
PCR give 100% accuracy?
Because of that perfect gene
If
we
we,
have.
no, if we give 100% accuracy, na, easily we can find out the
No,
vaccine
sir, no,
also.
sir. It gives 97%, 96 to 97% accuracy.
Because it is
PCR,
monitored.
actually, Mr. Time and Salt, PCR only gave the 100% of accuracy. Am I right? If the PCR gave, if a PCR can possible to give 100% accuracy, na, why didn't you find out the vaccine against the COVID-19?
Hello.
Hello.
PCR is
Nice
Yes,
question.
sir. I give one example more. Yeah, before 15 day in Haryana, a health
Mm,
minister take
mm,
a corona vaccine. Now
Okay.
he is a COVID positive.
mm.
Yeah, that's why
Hello.
I am also asking with my Taiman.
that
Hello,
corona
Taiman.
vaccine.
Taiman, my question is simple. If the PCR can possible to find out the 100% accuracy, na, why you people sir didn't find out the vaccine against the COVID-19?
Because vaccine has lot of it has more side effects than advantages. That's why there are now we are, before the COVID-19 arrive, we have couples of vaccine, but it has side effects more. We have vaccine with the minimum side effect. That's why it take long time. We have corona vaccine and from lockdown also, from March also, but it
Mm.
require minimum side effect. That's why we are requiring more time. Are you getting it?
So
We
already we have the vaccine, but by
And
the vaccine,
Tanmay,
the side
It
effects
has
also
side
there.
effect. No side effect. We have to
Tanmay.
yeah.
I have also
Hello.
heard one more news that when COVID-19 has entered in your body, it's a replication or there is a change in the nature. It is changing its nature.
Yeah,
That's why
but
it's quite difficult to find the
when virus
exact solution of it.
Yeah,
just
COVID change by a
It's
virus change
an
in the
RNA
body.
pattern. Sir, its
Body.
RNA pattern is also getting changed.
hey, virus is a
Yeah,
living
different
thing.
body,
Just like
different change.
Yes,
hello.
yes. When it enters in your body, it performs some reactions and it changes its nature. Even its RNA pattern gets changed. That's
I
why
will
there
tell.
is a difficulty in pattern recognition. Yes, sir. Yes, Tanmay, sir. Please.
Yeah. Virus is living thing. We are also living thing. Okay. And
Yes, sir.
yeah. And if one people go in Rajasthan and someone is in Maharashtra, which person will individual have more chances of growing well? Obviously of Maharashtra because all facilities are
Yes.
available. Just like that virus also. We have very difficult time in summer because it's difficult to us to survive in summer. Have 50 degrees Celsius. The same thing happened with that virus also. Hello, are you getting?
Hmm. Yes, yes, I'm
Yeah,
getting.
yeah.
Just like we are living, we have we have difficult times in summer. Just like that virus also.
I got sir. Means you are saying that many people are living in many regions, in many circumstances, in many different environments.
Yeah.
That's why there is a different types of patterns in COVID-19. That's why
And
there is a
COVID
difficulty in getting the exact
where-
pattern of it.
yeah, where the virus where it found
Maybe
comfort,
your
it
virus,
will grow.
maybe in a region where you are producing vaccines, it's a
Yeah.
helpful for in that region, but in other region maybe it's harmful.
Yeah, and virus found its comfortability, nothing like
Hmm.
other. Just like
Right.
that, it's comfortable. When you
Yes,
are
yes.
comfortable,
Like
you will
like
grow.
Tanmay sir is Savle means black color, and Nikhil sir is white because he is
Yeah,
from Haryana and Tanmay sir is from Maharashtra. Is it?
yeah. Here temperate region. The temperature is going about 50 degrees Celsius. That's why.
Very good. Yes, this is the exact reason, right?
Yeah.
Yeah, Nikhil
Just
sir
like
is right in that it's a yeah. This is the reason that it's not having the same pattern in every region, and even
yeah,
there.
and that's why
Actually, I have heard the
difference
different
in
that
comfort.
it's a changing its nature as well. That's why there is a
Yeah,
difficulty in yes, which type of
uh,
medicine is suitable for it.
we have
Yes, sir,
temperate region. We
definitely.
have many difficulties in there, but in Australia there will they will grow. They have many height, more height than Indian player. Did you find it that Australian players have very healthier and heighter than us? They have great height than us because they are comfortable there, so they are growing well. And as per our region, or our temperature or our nature, Maharashtra, we have that same pattern of Haida. If Indian people, if there are 10 peoples, 10 Indian peoples, and there are 10 peoples of Australia, we can easily recognize them because they have different nature. And the who one have best nature will grow nicely.
So, sir, I ask one question, Tanmay sir.
Hmm.
In the world, only Chinese people are powerful, or immune system is powerful? Because China is prevent
Nice
their
Uh,
question. Nice question.
citizen from COVID-19.
that its immune system is depend upon exposure to antigen. The China have discovering many more viruses, and they are eating wild animals. So there are many antigens present already in the body of the Chinese
Oh,
people. So they have developed that immune system. We have one. I will tell you that one we have memory T cell that helps to produce antibody in our body. So I think they have many more memory T cells. It is in 12th biology, memory T cells. Memory means if one antigen comes in our body, just like did you know about that chicken pox?
Yeah.
Chicken pox happens in like one only in lifetime.
Yes, sir.
Sir, Mata Rani.
I
Sir,
think
Mata
we
Rani.
all are facing, because I also suffering.
Yeah, but only
And
one time,
Tanmay sir,
each time.
Tanmay sir.
Yeah.
Hello, Tanmay sir. It's
Mm.
known as Mata Rani in North India.
Matter. Yeah, this happen only one time in our whole life. Anyone can know. I will
Yeah.
tell you.
Yes, sir.
Yeah,
Gautam sir, you
because
are also suffering in
Yeah.
your
In my
life,
school. Yeah. In my
everyone,
life.
everyone
and
suffer once.
mostly in childhood, mostly.
Yeah. Childhood. Childhood.
Yeah, everyone is suffered, but because of that memory cell, it can't happen second time. Because
Hmm.
memory cells itself tell that it has the memory of that virus,
Okay. Okay.
and memory means antibodies, and
Yeah. I
it
know
know the
that our human body
Yeah,
already
it's
take
like a
one
root
scan
memory.
copy.
It's like a
yeah,
root memory means once our body
Yeah.
got the
I'm also
I-
studied whenever affect on some disease now
but
already our body take one scan copy and one Xerox also.
can copy,
yes,
Xerox
so
copy,
even so
and certificate.
in mathematics, like if we have solved
Yeah.
a
Yeah.
question,
Yeah.
then we
Yeah.
can solve the similar
If we
questions, many similar
if
questions.
we know the
Yes.
formula, can, but if we rotate the question, we can't
Mhm.
solve. Just like virus is doing, virus is
Okay.
mutating, so that's why infection occur twice of
Okay.
COVID.
Okay.
If someone is getting ask me question of that, COVID is happening second time also. The answer is that the virus is mutating. That's why our body is unable to detect.
Okay.
Mutating is just changing, just like our professor rotated the question, and we can't get answer, even if we have memory of that question, because that question is rotate or change. Virus
Mm.
doing the same thing.
Okay. Okay.
Okay, are you satisfied or I'm just giving answer?
Yeah.
I know your mathematics is weak, but we can understand.
Yeah.
No,
I understand.
Sorry,
no,
but
not.
we are really now confirmed. Now, Brahmananda sir, please ask questions. Only Tanmay is speaking so far.
Hello.
yes, hello, sir.
Yes,
Uh,
welcome.
Actually, I have a query. I already asked him.
Okay.
Hello.
Any extra question? Yes, actually, sir, please take him
Yes.
test. Sir, please take his test. Next year he will become doctor.
couple of years, not just next year. Couple of years.
Sir,
Couple of years.
because
So,
1 year
yeah,
for
couple
training
of years.
also.
I
Yes,
have
Nikhil.
Yeah, I say 1 year for training also.
yeah. I have 2 more years to go.
Oh,
Okay.
you are a finalist student right now.
Yeah, final year. 1 year
Okay.
of academic year and 1 year of intern.
Oh, okay. 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 Which part?
That huh?
Specialist? Which
No,
specialist?
Means
for
Tanmay, you are
Veterinary doctor.
Okay.
veterinary.
Oh,
Tanmay,
okay.
Tanmay, you are fifth year student.
No, we have only 4 year, and fourth year is of
Okay, 1 year training. 1 year training. Okay.
Mm.
uh,
Village training.
Mhm.
yeah, village, or we have 6 months of Maharashtra tour and 6 months of other state tour.
Oh, okay. Okay. Okay.
Uh, 1 year
Mhm.
of 1 year of the training period, 6 year
Okay.
training period in Maharashtra and 6 year in
Yeah.
other states.
Hello, Tanmay sir.
Yeah,
When you want a government job for veterinary doctor, so you also give exam.
yeah. We have departmental exam for NPC, livestock development officer.
Yeah.
Yeah,
Okay.
in your every village or every town have LDO or veterinary dispensary.
Okay, sir.
Even in every village also.
Yeah, even every village also there is a post that is LDO, livestock development officer, where
Yes.
veterinaries are recruited at that post through NPC. MPC or other
Yeah.
state or other other exam. Maharashtra have Maharashtra Public Service.
Wow. Doctor
Sir, so
Sahab,
you
one
are
more question.
hello, Tanmay sir.
Yes,
So you are from particular Vidarbha.
yeah, Vidarbha,
Okay.
Nagpur,
Yeah.
Nagpur,
Okay.
Orange
Okay.
City.
What's your future plan? You wanna start a one clinic or you wanna serve with village side?
Yeah, village side.
Village
I
side.
want to give.
Yeah, after getting job, he will directly move abroad.
No, no, but only giving service to India, Atmanirbhar.
Oh,
Wow,
okay. Let's see
super.
after 2 years what will happen.
Giving service, giving lives to only Indian animals.
Okay. Let's see the
Sir,
same way repeat. Everyone understood you are committed. Okay.
one more,
There is a
sir,
Then
one more
let's
question.
see after
Sir,
two
one
years
more
what will
question.
happen. Yeah, everyone noted your words. Yeah.
Doctor
So
Sahab,
yeah.
Doctor
yeah,
Sahab, one more question. Tanmay,
Hmm.
yeah.
I did not see any news that animals are affected by COVID-19. What is the secret behind it?
Not so some there is an animal binding protein that coronavirus attached that if coronavirus has to enter in our body, it has to
Hmm?
bind animal binding protein.
Okay.
Hmm.
And what's regarding birds?
Huh?
What
Yeah, I'm
birds?
going to tell
Birds.
you. First, I have to complete my what.
Hmm.
There is animal binding protein. If the animal binding protein is present there in the animal or any species, and COVID-19 get attached, then it has COVID infection. If not, then the virus get excreted. So, if animal binding is protein in particular species or particular animal, if somewhere have infection of coronavirus in animal, there is animal binding protein where it get attached.
Okay. Okay. Okay.
Just like our gatekeeper. Hello. If you are passed to go in a program. Hello.
Hmm.
Hello. Hello.
Hello.
If you have one ticket from
Okay.
Amravati to Nagpur or Nagpur to Amravati, you
Hmm.
can go in any bus
Okay.
just like that.
Okay. Okay.
If
Is
the
there any sense COVID affect on plants as like rice?
hello, what?
Is the corona only affected wild animals and or is there any possible to affect on plants also? I mean plants that means rice, that kind of foods also.
Yeah.
They have
Plants or
no
birds.
particular virus get affect to the particular species. Plants have different genome from animal,
Yes.
from
Okay.
human.
So coronavirus mainly required a respiratory system.
Yeah, because they develop into respiratory. Yeah. Just like aquatic animals live in river or sea,
Okay. Hmm.
flying animal, land animal on land. Just like coronavirus in
Okay.
developing respiratory tract so that it
Oh,
surviving respiratory tract. And that question was the earlier question. Oh,
Yeah, in
which one?
birds.
Yeah, but because then, coronavirus first, previously it was of human, no, not of human, it's only affect animal, but it get muted and it changed its sequence so that that sequence not attached to the animal. Now it's attached to the human because the virus is compatible with human body.
So
And
scientists not found what is the origin of COVID-19 or
origin
coronavirus.
still there is COVID-19. If COVID-19 there is still discover not able. WHO is also not able to discover origin of coronavirus.
Okay.
But after every 100 years, there is a
Mm.
in 1820 there is a plague. In 1920 Spanish flu.
Yeah.
Yeah,
20
yeah. So
and
what is sir? Why swine flu also?
what?
Swine flu?
Hmm.
Yeah,
Swine
it
flu.
means yeah, swine flu. Those are from a
Yeah.
pig. Yeah, yeah. Effective from pig. And so swine flu are means week after means season are changed when summer are came.
Yeah, because how can I tell you? Mm,
But
that's it.
COVID-19 are strong in all season.
Yeah, because it mutate and coronavirus mutate so that it can survive in all. But my point of view, I think that it is prepared in lab. So that's why it is 24 into 7 into 365 days. I think so. If it was of nature, it was particular to the season. I think it was prepared in lab. And I think that because China is very cheapest country.
Oh, so COVID is the most comprehensive than AIDS, am I right?
It is artificial. If it is of natural, it is for a particular time period.
Okay.
Every
Okay.
natural is you can see now, raining is for only 3 months. Not now. Now
Mm.
it's all year, but raining is for particular time.
Mm.
Uh, Summer is for particular time. If
Mm.
it is natural,
Sir, so
if
COVID came from bottom of the earth means patalo.
no. I think it was prepared in lab. I was telling that answer that it is present all the summer, winter, and rainy season. I think it is artificial. If it is of a natural means, it was only for a particular period, just like swine flu, come and go. It
Okay.
is
Yeah,
artificial.
sir, but
No,
in Spanish flu survive for 3 year, 2 or 3 year.
3 to 5. It takes 3 to 4 years. Yes.
Yeah.
Lifetime is
Yeah.
14 days, am I right?
Actually, coronavirus is
Sir,
of 2000. It takes 18 times to make it powerful itself.
Okay.
It was just like a hibernation 18
Mm.
years. Then it
Mm.
come up with a strong power.
Mm. Mm.
If you have 6 hours of sleep or 8 hours of sleep, in the next day you will get feel powerful. Coronavirus of 18 years of night, so that's why it is more powerful. I think it takes 18 years to build that coronavirus or 18 years to mutate. Uh,
18 years.
yeah, because coronavirus origin of coronavirus was of 2002.
Mm.
Uh, it
Okay.
was in it was
Okay.
in poultry bird.
If someone recovers from corona again after 18 years, there is a chance to getting again corona positive.
No, actually it mutates coronavirus. It make a cell it self develop.
Mm.
Then it comes up and it
Mm.
is more dangerous. It was
Okay. Mm.
I think it was in particular animal
Sir,
which
yeah, I also
like
listened to first coronavirus in Canada lab
what?
in Canada lab
Uh,
and China took away from.
I don't know about this, but I think that coronavirus was prepared in lab
Yeah, definitely.
from such result because it is not.
Hello,
Hello.
sir. I think that's all for today. It's more than sufficient.
Yes, sir. Yes, sir.
And sir, if we are all having further queries, so we can continue the session for tomorrow as well. But
Okay.
I think it's more than sufficient. Yeah, if there will be any
Yeah,
questions,
it's
so
sufficient.
Nirmay is here. Siddharth sir, is it sufficient, or you want some more?
No, no.
I'm from after 7 December. I will all free because my exam will be over. The
Oh.
Oh,
offline
Okay.
exam is online, and
Okay.
after December,
then we will take your interview. Don't
yes,
worry.
Mm.
no. It will be a qualitative test.
Okay, nice. Okay, so I think that's all for today. And
I hope I answer all questions rightly or properly, or I satisfied all the answer. I hope
yes, all
Yeah,
you're all
satisfied. You did the
you're
best
all.
today.
yeah, thanks
In
for
the
your
starting,
patience also.
in the starting, you were saying that I can speak only about 10 minutes, but you completed 1 hour.
Yeah.
It's not a thing. Really, a single person on the other hand, six members. You
Sir,
are
I
speaking.
think Tanmay, hello. I think Tanmay, sir, have lots of experience in public speaking in his college also.
Really, he's a very nice public speaker. Even you will not feel bored. It's
Oh,
not a boredom. It's a interesting thing. He always
Oh,
speak.
okay, okay, okay.
Even whenever I used to add him in a what we lecture topics,
Mm,
I was immediately adding him, and without preparation, he used to speak around 10 minutes without preparation, without knowing the topic.
okay,
And it's
okay.
the best, yeah, really, it was the best content whatever he was speaking. Even
Oh,
I was searching on Google and I was trying to find anywhere, but
as
really,
like
he was.
Brahmananda.
yes,
I
sir,
think
yes, sir.
Brahmananda is a really good speaker, public
Yeah,
speaker.
Varmanan sir also speak. Very good.
Yeah,
Yes, yes, right.
yeah.
Okay, sir. Thank you very much.
But today
I
Varmanan
was,
sir is silent.
because Tanmay is speaking.
Because of
No,
COVID-19.
because today's
So
Yes,
he
sir,
today's
leave
field
the
Varmanan
is
sir
place of
give guard.
Tanmay.
no, today's field
Very
is
Today he
related.
leave the place for Tanmay.
Yes,
No, no,
yes.
nothing like that. Today's field was related to medical, so that why. If
Yeah.
tomorrow topic will be engineer on engineer or maths, I will be
Uh,
silent.
yeah.
No, no, Varmanan sir speak in all matter. He
Yeah.
have
He
knowledge.
is the
All
encyclopedia.
around,
He is
all
the
around.
all-rounder part
Yeah,
of
he is
Hardik,
the encyclopedia.
Hardik Pandya. Just like
Okay,
Hardik Pandya.
Brahmananda sir, one compliment. Brahmananda sir, closing sentence. Hello, Brahmananda sir, kindly unmute and give closing sentence. We will end.
Yes.
Yeah, it's very cold, I know. Hello, Brahmananda sir, where are you?
I
Yes,
think
sir. Yes, sir. Sir, hello.
Yes, yes, sir, please give closing sentence.
Sir, this is enough. Actually, this is enough. If you all people will make lots of admiration with my name, I'm not enough for that. And what I will say, this is a really euphoric discussion today, and I get lots of knowledge. I extract lots of knowledge from here. Thank you so much. What this lecture? Thank you, sir.
Mr. Tanmay, Mr. Tanmay.
So
Yes,
then,
Sagar,
sir.
I was little bit nervous that I will able to speak in English or not because there was large gap between previous call and now. As
I can
Sagar knows,
understand this, but you are ultimately doctor, no? No one can
No,
understand
sir.
the doctor.
Yes, sir. No one can understand the doctor writing, but
Yes.
writing every person can understand. And you are a natural speaker, I think.
no.
Yeah.
Sagar, because of Sagar sir, I have developed
Mm.
this quality. I think because
Thank,
Right.
since from I think we have just only two people's. Yeah, Sagar, we
Yeah,
have
but
started.
I'm also connected with you around more than 1 year. Yes, it's been more than 1 year. I have connected
Yeah,
with you.
that's why I'm not a public speaker, but because of Sagar sir, I have and now I have a lot of confidence because
And
of
what
Sagar.
about me? I'm not a speaker at all. Yeah, really a big, really very nice speaker. Really, Tanmay, super.
I have learned many things of Sagar sir also.
Ah,
Yeah, I'm
Because
also
it is
developed my skill because of Sagar.
yeah
Earlier,
array,
my
because
array.
English is also very
It's
poor, but nowadays
it is
I'm also
it's
learn little bit in
it's
English.
no, no,
it
no,
has power
no.
it has power of connecting people.
Yeah, yeah.
I'm
Sagar,
Yeah,
close to this one. It's a hyper. There was one figure of speech. What is the name? Um, Sorry, I'm forgetting. It's so overstating me. I cannot accept.
oh,
no,
that's
no,
fine.
sir. Really, I was also lots of time told you because
Yeah,
of your
and
curiousness and your eagerness. You make this group. This is a help the all people a lot.
Yeah.
And yeah, Grateful for you, sir.
I have one Mara for
Thank you
Sagar
very
sir.
much, and that's all for today.
Yes, sir. Thank you. Thank you so much. Good
Yes, sir.
night.
Thank you to all